we're seeing in this picture is a view of our blood. Our blood is a very important component of our body's transportation system, and we're going to take a closer look at it here. Now, our blood is considered a complex connective tissue, and as a complex connective tissue, it does have both some living and then also some non-living components. If we were to take a sample of our blood and we were to prepare it in a centrifuge, that's a special instrument that spins samples very quickly, and it separates samples depending on their density. If we were to do that, we would see that our blood is composed of layers, so components that do have different densities. We have the blood cells. Those are what we commonly refer to as the formed elements, and we would have both red blood cells and white blood cells. And then we also have what's called the plasma, and that is the matrix. If you look in the image right here, you can see that we do have primarily red blood cells. Those are going to be the primary formed elements that we have, and that might be about 45% of our blood. And then we also have the plasma making up the largest portion of our blood, and 55% is what's shown here. And then we have a very thin layer in the middle, which we call the buffy coat, and that is composed of leukocytes and then also platelets. The blood, if we were to take it and put some drops of the blood in water, you'd have something like you see in this image right here. And that's because blood is going to be heavier, it's thicker than water, so you can see that it does sink in the water, and it's also slightly alkaline. That means that the pH is a little bit on the basic side. So the pH is between 7.35, 7.45, so somewhere in there, just slightly alkaline. And the temperature of the blood is 100.4 degrees Fahrenheit. This is just a closer look at the different layers of the blood or components of the blood. If we talk a little bit more about the plasma and what's actually in the plasma is primarily water, but then dissolved in that water, we have a variety of different electrolytes or salts. We have different plasma proteins, and then you can see that there would be other substances that will be found in the blood because they're going to be transported in the blood. So those are going to be found in the plasma. And then the formed cell, we have erythrocytes, which are our red blood cells. We have leukocytes, which are going to be white blood cells. And then we also have the platelet component. We talk a little bit more about the formed elements. The formed elements, we said that this is going to be the blood cells themselves. We can see some of them in the image right here. We have erythrocytes, which are red blood cells. We have leukocytes, which are white blood cells. And then we also have platelet components. The erythrocytes, these are the most abundant. So that's most of the cells that you're seeing in this image right here. Their primary job is going to be to carry oxygen to other cells. So they will have special proteins that help them to carry that oxygen around our body. So here we have a look at the red blood cells, or what we refer to as erythrocytes. These have a very characteristic shape to them. They're quite flat. They have that concave or depressed center in them. And then some other characteristics. One of them is that they're going to be anucleate. That means that they do not have a nucleus inside of them. And they're also going to contain relatively few organelles. So very few organelles are going to be in there. And one of the reasons for that is that they have this big abundance of hemoglobin molecules. There are over 250 million hemoglobin molecules in every single red blood cell. So those are taking up the majority of the inside of the red blood cell. And the job of the hemoglobin is going to be to carry the oxygen, which is the primary job of the red blood cells. This specific shape that we have to the red blood cells, their small size, their depressed centers, this is going to allow for a large surface area, and that large surface area relative to volume is going to be good for the transport of oxygen. If we look at some abnormalities that can happen with regard to our red blood cells, anemia is quite a common one. Anemia is going to be any condition where there happens to be a decrease in the oxygen carrying capacity or ability of the blood. And this can result from a couple different things. Typically, this is going to be either a lower than normal number of red blood cells, because they are the ones that are carrying the oxygen, or it can be an abnormality or deficiency in the hemoglobin content. Hemoglobin is the specific protein that's filling the inside of our red blood cells that's actually going to be binding that oxygen. So if we do have a decreased amount or just abnormal hemoglobin, that would cause us to have less oxygen being carried in our blood. Now, a specific type of anemia is sickle cell anemia. And sickle cell anemia, it is a genetic condition. So it's hereditary. And this is because of the actual composition of the hemoglobin molecule. The DNA sequence is going to be a little bit different for determining the instructions to make this particular protein. And so if the instructions for making the hemoglobin are different, then the hemoglobin that we do make from those instructions is going to be different. And you can see the difference here in the slide. It's showing you on the bottom in the purple there, that is the actual protein sequence or amino acid sequence that makes up the hemoglobin. And notice that when someone has this condition, they have this one amino acid that's a little bit different from what it ought to be. And that results in an overall difference in the three-dimensional structure of the hemoglobin molecule. And the end result is that the red blood cells do get shaped a little bit differently. So they have more of a sickle shape, which is what we're seeing in the image here. And so what this is going to do is it's going to cause the red blood cells to rupture easily. They dam up when they're moving through the small blood vessels. And this is going to happen when the oxygen content of the blood is low. Okay, so that's sickle cell anemia. It is a genetic disorder, and it is something that is inherited in what we call a recessive manner. This condition, polycythemia, this is going to be when there's an excessive increase in the red blood cells. So you can see the comparison here. The first image is going to be what our blood composition looks like in a normal individual. The second one would be perhaps what you have when you have anemia. So notice that there's a small number of red blood cells relative to normal. And then in this condition, you have a whole lot of extra red blood cells. So polycythemia is going to be when we have extra red blood cells, an excessive increase in the red blood cells. The leukocytes are going to be our second type of blood cell. These are the white blood cells, or WBCs. And you can see that in the image right here. So they're going to be larger. They're going to have a more spherical structure to them. They are less numerous, which is also emphasized in the image right here. Now, instead of actually being responsible for carrying oxygen to various tissues, these play a crucial role in defense against diseases. They contain nuclei and organelles, which is again a difference compared to the red blood cells. They are going to be able to slip in and out of the blood vessels. And that is something that they're going to be doing when they are involved in that whole defense response. Lastly, these are something that's going to become more abundant upon infection. So when someone does have a higher white blood cell count, then there is suspicion of infection in those specific cases. Leukemia is an example of a condition where we have very high numbers of white blood cells. So typically the white blood cells are relatively low, which is what we're seeing in the image right here. Normal blood is going to be mostly red blood cells with some leukocytes inside of there. But when we're looking at leukemia, notice that there is this vast increase in the abundance of the white blood cells. Now, these white blood cells, it might sound like it's going to be a great thing if we have extra defense cells there, but they are immature white blood cells, and they're not going to function properly. So these extra white blood cells that we have in the case of leukemia, they actually attack the body, which of course is going to be a detrimental thing. When we look at the leukocytes, there are different types of leukocytes. So this is showing you the way that the leukocytes can be classified. They have different names. They have different ways that they appear. They have different functions that they play. The leukocytes themselves, they are divided into two types. We have the granulocytes, and then we have the agranulocytes. If we look at the granulocytes first, there's three types of these: the neutrophils, the eosinophils, and then we also have the basophils. The neutrophils, these are going to be the most numerous of the leukocytes. So these are our most numerous white blood cells. They are going to have a multi-lobed nucleus, which is a little bit different, a little bit unusual. Their job is going to be to attack bacteria and also to attack fungi. They are avid phagocytes. And when we're saying phagocytes, what that means is that they can engulf and kind of swallow up those bacterial cells or those fungal cells that they do come into contact with. So that's the neutrophils. The eosinophils, these are going to be responsible for digesting and attacking parasitic worms that we might have in our bodies. These specifically contain lysosome-like granules that would do the digestion of those worms. And then the last type of granulocyte is the basophils. These are the rarest of our white blood cells, and they are not really responsible for attacking pathogens that we might find inside of our bodies, but instead they are attracting white blood cells to inflamed sites. So if you have inflamed areas, these would be kind of calling out and attracting those other white blood cells. The granulocytes that we see here at the bottom, these have two types. We have lymphocytes and then also monocytes. And the lymphocytes, these are the second most abundant white blood cell. So first is going to be the neutrophils, then we have the lymphocytes, and they have a major role in the immune response. The monocytes, these are the largest in size. They have a U-shaped nucleus, and they also have the ability to change into macrophages. Macrophages would be swallowing up or kind of eating and consuming different pathogens. The platelets are the last part of the formed elements. The platelets are unusual because these are not cells, and since they're not cells, we have a different shape to them. They're just like little fragments of multinucleated cells. Again, we said they're not cells because they're just fragments of the cells, and we call them megakaryocytes. So you can see that they have odd shapes in this slide right here. We can see we've got the red blood cells. We've got white blood cells in here. And then all of these little pieces right here, these would be the platelets. Okay, so they have a very unusual structure, shape to them, and these are important for the blood clotting process. So they're needed for normal blood clotting when the blood vessels happen to be ruptured or if the blood vessels happen to be broken for some particular reason. Hematopoiesis is blood cell formation. This is something that happens every single day in our bodies, and specifically this is going to happen in our red bone marrow or our myeloid tissue. The number of cells that we produce using this is really tremendous because every single day on average we produce 100 billion new cells. There are going to be specific stem cells called hemocytoblast. These can develop into different types of blood cells. You can see right here that some of them will develop into lymphocytes. Some of them will develop into first a myeloid stem cell, which develops into a variety of different cell types. And so depending on the needs of the body, then we have different types of cells that are going to be produced from that initial hemocytoblast stem cell. If we look specifically at red blood cell formation, red blood cells live for approximately 100 to 120 days. So since they only live for really a few months, they do definitely have to be continuously produced. Since they are anucleate cells, cells that don't have a nucleus, this means that they're not able to synthesize proteins, they can't grow, they can't divide, so a single red blood cell cannot produce additional red blood cells. And for that reason, we have to go back to the hematopoiesis process, that red bone marrow, if we need to produce new red blood cells. The lost cells are going to be replaced continuously using that process, so from the stem cells. So we will have specific stem cells in the bone marrow. They will produce the red blood cells, which are going to initially have a nucleus. That nucleus is going to be lost over time after we have the hemoglobin produced inside of those cells. The production of the hemoglobin is going to give the cells their characteristic red coloration as they're carrying the oxygen, and then they will enter the bloodstream. The job of the red blood cells is going to be to maintain homeostasis of normal blood oxygen levels. So they have the hemoglobin. The hemoglobin is going to be carrying the oxygen around, and they are responsible for responding to any change in oxygen levels to help maintain that homeostasis level. So if there happened to be some type of stimulus that indicated there was low blood oxygen, this might be a decrease in the red blood cells, a decrease in the amount of hemoglobin, just decreased availability of oxygen. What that's going to do is that's going to stimulate the kidney, the liver in some cases, to release erythropoietin. Now that right there is going to go and it's going to stimulate the red bone marrow. And when we stimulate the red bone marrow, we're going to be stimulating it to go through the process of blood cell formation. So we will get production of erythrocytes, the red blood cells that are capable of carrying oxygen. And so in that case, when we get production of more of those, we would expect that we're going to get the normal oxygen levels returned to what they should be. So that's an example of how the red blood cells are going to function in homeostasis. Specifically, in this case, they're functioning to control oxygen levels in our body. The production of white blood cells and platelets is also going to depend on hormones. We're just not specifically looking at the production of those in this particular slide. If we talk about hemostasis, this is going to be clotting or stoppage of bleeding. There's a number of different steps that take place when we're looking at stoppage of bleeding. If we review those here, the first thing is that in step 1, there have to be some vascular spasms that occur. And what we mean by this is that the smooth muscles are going to contract, and when they contract, they're going to narrow the blood vessel. So that is what we see happening in the first picture here. In step 2, we're going to have a platelet plug that's formed. Platelets have a big role in the clotting process. These platelets, we're going to have them formed when, first of all, we have an injury to the vessel lining that exposes collagen fibers. Once those collagen fibers are exposed, the platelets are going to start to adhere to that, and the platelets themselves release chemicals which cause the nearby platelets to become sticky. So we start to have this clumping together of platelets, and we get the formation of that plug. Once we have the formation of that plug, then we get the coagulation process begin. This coagulation process is going to involve joining of fibrinomolecules to fibrin, which forms a mesh, and then that mesh is going to create the clot, which will eventually stop the bleeding. So that is hemostasis that we're looking at right there. This would be something that happens if there's an injury to some type of blood vessel and we start to have some leakage from that blood vessel. Now here's just a closer look at it. In this picture, we're seeing the mesh that would be forming to create that actual clot. There are a number of clotting disorders that can take place if there's an error in this overall process. These clotting disorders, they can result in a number of different errors in the process, but we get a thrombus, which is going to be a clot that develops and it persists in an unbroken blood vessel. Now, sometimes that thrombus is going to kind of stay in position. Sometimes it actually dislodges and then it moves. So if it does dislodge and then it moves, it's called an embolus. And that embolus can get caught if we do have it move into a smaller vessel. Bleeding disorders are deficits in clotting factors. So this would be if the clots don't form the way that they should. And this example, thrombocytopenia, is an example of that. This is when we have insufficient platelets, and platelets play a big role in that overall clotting process. So they're supposed to clump up and form that initial plug. In this condition, we don't have enough of them. You can see in the pictures here the difference between, say, normal platelet counts and low platelet counts. The platelets are the little yellow pieces that we see right here. So in normal blood, you can see that there's quite a few of them in our image. If we have a low platelet count, you can see that there's just a couple of them there. So this causes a problem with clotting. The clotting doesn't happen like we would expect. Hemophilia, there's a variety of disorders there in that group, but these result ultimately from a lack of clotting factors. If we talk a little bit about blood groups, our red blood cells, they do contain genetically determined proteins on the exterior, and we refer to these as antigens. As antigens, these are substances that are recognized by the body as foreign. Specifically, they are recognized by antibodies, and antibodies, they have a very specific recognition of antigens. So looking at the images right here, you can see that this red blood cell, which is what this is showing us, this red blood cell, it has these different antigens sticking out. So this is the antigen. And that antigen would be recognized by specific antibodies. The antibodies are the little Y-shaped structures right here. So that's the antibody. And so if there is a specific recognition between them, then you'll have this kind of binding together that we see down here in the bottom. In the top picture, there's no recognition because the shapes do not match, but down here at the bottom, they do match. So when they do match up like that, and that antibody can recognize and bind to that antigen, what that causes is a clumping up of these red blood cells. That clumping up is what we call agglutination. So agglutination is happening down here. So clumping, because the antibodies recognize these red blood cells as being foreign. And then the picture at the top, these would be no recognition. So if there's no recognition, we're not going to have clumping of the red blood cells, or we would not have agglutination take place. All of this is important because this does affect the whole blood transfusion process and what type of blood we're able to receive, or what type of blood we might be able to donate to someone else. Essentially, our body is going to recognize things that are different from what we have as being foreign, and we have antibodies that would match up with that, and they would cause that agglutination process and clumping. If you are someone that has type AB blood, then your body is used to seeing the A antigen and the B antigen, and we don't have antibodies that are going to recognize and bind specifically to those. So that means that any blood that had the A or the B antigen on the outside would be okay for someone that has type AB blood, so they can receive that. If we're talking about somebody that has type B blood, for someone that has type B blood, type B would be something that would be normal. There's not going to be antibodies that match up specifically with B, but instead we do have antibodies that would match up with the A antigens. So notice that someone that has type B blood, they're going to have the anti-A antigen, and that means that if they did get blood transmitted to them that did have the type A antigen, then they're going to recognize that as being foreign, and that's going to cause the agglutination process. So someone with type B blood cannot receive anything that's A. They can't receive type A. They can't receive AB blood either. Someone with type A, they can receive A, but they can't receive anything that has B's on the outside because they happen to have the anti-B antigen, and they would recognize that as foreign. They would bind, and we would get the agglutination process. Someone with type O blood does not have as many options for receiving blood because they have anti-A and anti-B antigen, which means that if there's any blood type that has A or B in it, that is something that their body would recognize as foreign, and we would get the agglutination process there. Here's just a look at how blood types are actually determined. What they do is they basically take the blood and then they mix it with anti-A, and they also mix it with anti-B, and they look for the agglutination process. So if there's any A present, then we are going to get agglutination, which in type AB individuals, we would expect to get some agglutination with anti-A. We would also expect to have some agglutination with anti-B. If someone is type B, then notice that there's no agglutination with anti-A because there's no A's there to recognize, but we do have agglutination with anti-B. And someone with type A, they just have the agglutination or the clumping with anti-A. And then someone with type O blood, there's not going to be any agglutination with the anti-A antibodies or the anti-B antibodies.